The overall aim of the following experiment is to record and characterize human infant nociceptive specific brain and spinal cord activity. This is achieved using EEG and EMG techniques to measure electrophysiological activity in the central nervous system following clinically essential noxious procedures. The first step is to ensure that high-quality physiological recordings are obtained from infants when they experience experimental tactile and essential noxious stimuli. Next, post-processing analytical techniques need to be applied to the data in order to characterize the evoked patterns of activity. Results can be obtained that show nociceptive specific brain and spinal cord activity can be recorded from the human infant brain.
This method will help us understand how the developing nervous system responds to noxious stimulation.
To begin setup for this experiment, first prep the infant's skin, and then place a minimum of 16 individual disposable EEG silver chloride cup electrodes on the head according to the modified International 1020 electrode placement system. Use EEG conductive paste to optimize electrode skin electric coupling. Here, a schematic of electrode placement for EEG recordings can be seen, modified from the International 1020 electrode placement system. Use FCZ as the reference electrode for the recording. Use the same ground electrode for the ECG and the EEG. Place a ground electrode onto the chest or head. Then,